Number 3. So this one is, I apologize, it's a bit of a painful lecture. There is a lot of memorization and there is a lot of getting used to the cells and what the cells are doing and the personality of the cells and all those things, how they derive, how they develop. But of course, we're only touching the surface because the whole course has to do with functions of the cells, right? So here we're just going to start touching the surface on the cells development and function. Okay. So main ideas here is that you start from a pluripotent stem cell. It's also called a hematopoietic stem cell. Okay. And the stem cell can differentiate into all the different cell types. So the hematopoietic stem cell in your red bone marrow is going to decide which cell is going to become based on what your body needs. So you have cells that are derived based on one certain path, that is like the myeloid path. So most of your innate immune system cells are derived from the myeloid path, as you see. Okay, and what that means is that most of those, including here the natural killer cell, this one is from the lymphoid path. So your innate immune cells, they all come with their already made receptors on their surface. Okay, some of these cells even have receptors inside of the cells, not only on the surface. Okay, but they come with the receptors that are already coded by the DNA that is present from before birth. Okay, so this is why you call, we say that they have pre-existing receptors. Okay, nothing genetically modifying is going to happen with those receptors. Okay, and when you compare those to your adaptive immune cells, when you say adaptive, you mainly think B cells and T cells, right? So the receptors on those cells, those are the receptors you're not born with. You have to develop the receptors as you go along, okay? So these cells are going to undergo a lot of gene shuffling. So genetic shuffling is going to happen with these cells. And you are going to start developing those cells as you are exposed to certain antigens. So if you're not exposed to certain antigens, then you're not going to develop a specific, more mature type of adapter B and T cells. Okay? Yeah. So to make the learning of these cells characters easier, I did put here these 2 links. This is just a trailer for these cells at work. And here is like a whole movie, I think it's about an hour and a half or so, telling you the story of this, scenario on these cells that are working in the body. So it's telling you the story of this little red blood cell that is trying to get to the lungs and get oxygenated. And it keeps on running to places it's not supposed to go. It's actually a cute cartoon thing, if you enjoy cartoons. And it's quite accurate too. And of course, they have bad guys, or those are like the bacteria, something like that. Okay. All right, so here is a big picture of your formed elements in the blood development. So formed elements, you have the cells, and all white blood cells, red blood cells, and also platelets. Okay, so you start here with your multipotent or pluripotent hematopoietic stem cell. So this cell here, considering there is a stem cell, this cell is self-renewing. Okay, so when you need to produce any of your final cells here, this cell here is going to receive a signal from the environment that it should divide. So when it divides, Here you have your potent stem cell. Okay. So when it divides, it's going to give two daughter cells. One of the daughter cells is going to be just like the parent cell. It's going to keep on being a stem cell. The other cell is going to become a precursor cell and is going to decide what is going to become later. Okay, so when you need to produce blood cells, you're going to have your stem cell that is going to renew cell and is going to give you a precursor cell that is going to be able to further differentiate. Okay? So this is all happening in the red bone marrow. Okay? And here is where you have the subdivisions of the two different paths. So this is from your book. So here you have one path that goes towards the lymphoid cells. So the common lymphoid precursor or progenitor is going to decide whether it's going to become a B cell or a T cell or a natural killer cell or a variation of a natural killer cell that is actually innate T cell or innate lymphoid cell type of progenitor. Okay, so these two types here that you can just group them into natural killer cells, these are the ones that function in innate immune response. And then the other cells here are the ones that are really, really important for your adaptive immune response. Okay, so that is your lymphoid lineage. Now, when you look here at the myeloid lineage, you see that everything else is deriving from the myeloid lineage. Okay. And yes, we are going to have some details on the differentiation of those cells. Okay. So here, this is just the following picture of the same, in the same 1.1 figure. Okay, so the previous slide was part A, so this one is like part B. So here you have your cells that came from the two different lineages, and after that, some of the cells are going to go into lymphoid tissue after it passed through the blood, and they are going to further differentiate, or they're going to be exposed to antigens that will be presented to them. And this cell here, like right in the middle, right between the myeloid and lymphoid lineage, this cell is very, very important. So this is your dendritic cell. The dendritic cell is very much a link between your innate and your adaptive immune system. Okay, so the dendritic cell is going to go from tissues that are not part of secondary lymphoid tissues. So they're going to travel from other tissues and they're going to go to those secondary lymphoid tissues and interact with your initial cells of your adaptive immune system. Okay, so dendritic cells are really, really important for that. Okay. After interaction with dendritic cells, you are going to further change your adaptive cells to become experienced cells. So before they interact with the presenting cells, they are not experienced, or you call them naive cells. Then after they interact, they become experienced, so they become effector cells. Okay? So here, looking at your B cells, they are going to transform themselves from cells that have receptors on their surface to cells that can shoot out receptors. And those receptors at this point, they are called soluble antibodies. Okay, so that's all the job of your B cell. So B cell, when it gets experience and becomes effector cell, it becomes plasma cell. And the plasma cell is the one that produces antibodies that go into the blood. Okay. Then you have this whole story with T cells. Okay, that's like quite complicated. But basically, the receptors on the T cells, they never detach from the surface of the T cell. Okay, so this is one major difference between B cells and the T cells. the T cell receptor stays on the surface of the T cell. But then the cell, after they see antigen, they also become activated, they also become effector cell, they can become all different types of effector cells. Okay. All right, so let's start slowly, right? So here is the story of your pluripotent stem cell. Right? Do you know some other type of stem cell? Like, yeah, when you think about stem cell? Yeah, you're thinking about embryonic stem cells, right? So the embryonic stem cells, those are types of cells that they can become anything, any tissue, any organ in the body, right? So those are the cells that divide and start to differentiate in all different types of tissues and organs. So you have a stem cell that is going to become is going to become a neuron, right? You have embryonic stem cell that is going to become the liver, another one is going to become the eye. So they can differentiate into all sorts of tissues, right? Keep in mind that all these stem cells, being embryonic or hematopoietic, they have the same genes, they have the same chromosomes. So all the cells in your body, except for gametes, right? And also except for mature B and T cells, right? All the cells in the body, they have the same DNA, they have the same chromosomes. So what differentiates whether a cell is a liver cell, whether it is a skin cell, is really how those genes are expressed, right? So if you compare an embryonic stem cell with a pluripotent hematopoietic stem cell, they have the same DNA, okay? It's just that these cells over here, they are part of the bone marrow, the red bone marrow. Okay, so these cells are not capable of becoming something else or some other tissue. They are really restricted in becoming cells of something that's going to be composing the blood. Okay. All right. Just as a side note, if you take out the hematopoietic stem cell, or they also call the mesenchymal stem cell, if you isolate mesenchymal stem cell from a person and you put them in a different environment that has certain growth factors, then you could potentially develop some other tissues in the body. Okay, so when you hear about stem cell research, well, automatically you think, oh, it has to be embryonic stem cells. And then you have a bunch of politicians that say, oh, no, forget about the stem cell research. Oh, stem cell research is evil, you're killing babies. That's not the case. Because if you are running research with stem cells that are mesenchymal stem cells, you can isolate the cells out of your own body, out of your own blood. Okay? So when you hear stem cell, don't automatically think that the scientists are baby killers. That's not the case. Right? But then, you always have not good. Okay. So back here to mesenchymal stem cells, right? So you mainly have two lines of differentiation for these guys. Okay, they're stuck in the bone marrow. Some of them float also in the blood, in the circulation, but the ones that are going to differentiate, those are the ones stuck in the bone marrow. Okay, they are there, they're sitting there, and they're waiting for a signal in order for them to start differentiating. First they divide, and then they're going to differentiate. So here, they're either going to take one path or the other path. Again, when you look here at the lymphoid path, you're going to see that you're going to have 3 cell types that are major for this lymphoid pathway. Okay, everybody else is going to come from your myeloid pathway. So what do? You memorize the ones that are coming from the lymphoid, because you only have to think about 3 of them, right? So here you say lymphoid pathway is going to generate T cells, B cells, and natural killer cells, right? Now, if I ask you something like, which path are eosinophils deriving from? And you're going to say, well, eosinophil is not T cell or B cell or natural killer cells, so it must be from the myeloid pathway. Okay, so everybody else is myeloid path. All right, so here is just in words what I have mentioned about your hematopoietic stem cell. They are self-renewed, right? So when they divide, one keeps on being a stem cell, and the other one is going to differentiate. And they are in the bone marrow. Okay, so here we start with a bit of details on how this differentiation is happening. So first, you look in the bone marrow and you have the stem cells that are sitting there and not doing much. They're not really metabolizing a lot. They are kind of like quiescent, right? So this is what you call the cells that are just sitting there for a long time, LT, right? Sitting there for a long time, they're quiescent, they're quiet, they're just sitting. So the term for those are long-term hematopoietic stem cells. They just sit there. Then let's say that something happens in the environment and the blood is circulating and there is a message in the blood, usually is either a growth factor or a hormone, right? So message comes to the bone marrow and it tells this long-term stem cell that it should divide and it should become a certain type of cell, okay? So then the stem cell, one of them is going to divide into two cells. One of them is going to stay there and it's going to also be long-term. The other one is going to become this short-term hematopoietic stem cell. So this guy here is metabolically active and it's not going to sit around there for a long time. It's a cell type that is in transition into becoming something else. Okay, so during this transition stage, depending on what information the original cell received, this cell here is going to start to upregulate certain transcription factors. So I'll say, don't worry about everything that is going on over here. There's a lot of stuff to know. Okay, so I'll say like, forget about these guys now. But there are some major transcription factors that are going to be upregulated. And this cell here is going to become this major progenitor cell. Okay, you call this guy here the multipotent progenitor cell. Okay, so it's like MPP, multipotent progenitor cell. Now, this guy here, when it becomes a multipotent cell, it is going to start expressing receptors on the surface that are different from the previous cells, cell types. So one main characteristic of a receptor that shows up on this MPP cell is this receptor right here that is called CD34. Okay, so when your precursor cell starts to express CD34, then it is assigned to become this MPP cell that is also in transition and is going to decide whether it's going to take the myeloid path or is going to take the lymphoid path. But then up to this point, the receptors on those cells, they are the same for both pathways. Okay? The cell is becoming more and more specialized on expressing receptors. Okay, so now let's say that this MPP guy wants to take the myeloid path. Okay, so what's going to happen here in the bone marrow, you have further messages that got there because it came from the blood, right? So some of these messages, those are hormones. Okay, so what is a hormone? A hormone is molecule that is produced by one cell in the body, by what type of cell, and then it is dumped. This product is dumped in the blood and travels in the blood and gets to the target cell. Okay, so here are examples of hormones. Most important ones here are erythropoietin. And what makes erythropoietin are really cells that are in your kidneys. Yeah.
Could you explain what the hormone is again?
Oh, okay. So hormone here. When you have one cell, so let's say here you have your kidneys. Okay. So here's your kidney kidney. So your kidneys, they have sensors here, and they are going to feel that your blood doesn't have enough red blood cell. So the kidneys are going to produce this molecule. This is a molecule. So here's a molecule. This molecule here is called erythropoietin. This molecule gets dumped in your circulation. This is your blood vessel. Okay, gets into the blood vessel, and it travels all the way, all around your body, and eventually is going to reach a target cell. What is a target cell? Is a cell that has a receptor for that hormone. Okay, so here you're going to get to your bone marrow, and in the bone marrow you are going to have this precursor cell that has a receptor. Here's a receptor that is going to bind your erythropoietin right there. Okay? So the hormone traveled in the blood, got to the target cell, and when it interacted with the receptor, it's going to send signals to the nucleus of this cell over here, that is MPP cell, and it's going to tell it to upregulate certain genes and downregulate some other genes, and it's going to tell this cell to transform into something else. Okay? But the hormone, the definition of a hormone is a molecule that is produced in one place and is dumped into the blood and travels in the blood to find its target. That's the hormone. You're welcome. Okay. All right, so here, erythropoietin is the hormone, is a hormone that is produced by the kidneys. It goes to the red blood marrow, and it tells the MPP cell that you need more red blood cells. Okay, so your precursor cell starts to upregulate certain genes that are going to transform this thing into a red blood cell. Okay, it's also called erythrocyte. All right. Now let's say that if your body doesn't need red blood cells, let's say that your body needs platelets. So there is a hormone for that. So there is this guy here that's called thrombopoietin. Thrombopoietin is not produced by the kidneys, is produced by the liver. Okay, so again, it's produced and is dumped in the circulation. It gets all the way to your red bone marrow and tells MPP cell that you need platelets. Okay, now your MPP cell is going to start to produce transcription factors, and you're going to end up with a megakaryocyte, and then you're going to get your platelets. We're going to talk more about that. All right, so besides that, so here you have like hormone for red blood cells, you have a hormone for platelets, and then you have these other hormones that they are stimulating factors, okay? So those hormones are really, really important for producing white blood cells. All right, so you have here your GCSF, you have GMCSF. So basically the G is for granulocyte, okay, granulocyte cell stimulating factor. Okay. So now if your granulocyte stimulating factor gets to your bone marrow, it stimulates your precursor cell to become some white blood cell or some granulocytes. Okay, so these are actions of hormones or growth factors that will tell your progenitor cell to become something else. Yeah. Oh, that is a very deep question. And basically, you have sensors in certain organs that will feel like, for example, when you are looking at production of EPO in the kidneys, let's say that your kidneys felt that you don't have enough red blood cells. So you have certain cells there that are like counting the number of red blood cells, and if they feel that there's not enough, so then those cells start to produce EPO. When you have enough production of red blood cells and your level in the blood reaches satisfactory level, then your kidney is going to feel that too. So it's going to stop producing EPO, so you don't keep on stimulating the production of more red blood cells. So there is this balance in the concentration. So you always have receptors in certain cells that are going to feel if your body needs certain types of cells or not.
Does that also work resources that kidney work? Does your kidney doesn't work, you don't have a kidney?
Exactly. That's not really for EPO. You need your kidneys to work for that. So if you have kidney failure, you are going to have problems of anemia because nothing is going to tell your red bone marrow to produce red blood cells. Yeah, that's one of the problems. I mean, the kidney has so many functions, but you have to inject EPO. Yeah, and there is also an interesting question because if you look at levels of red blood cells, in the body, you know that in general men have higher number of red blood cells, higher levels. Everybody took a AMP2? Yeah. Everybody in the class here took AMP2. So remember those. What's the name of that percentage of red blood cells?
Hematocrit.
Yes, hematocrit. Sometimes my brain goes blank. Okay, hematocrit, right? So if you have a bit of higher hematocrit count, percentage of red blood cells, then you know that your blood is carrying more oxygen around, and you know that you can deliver it more effectively to tissues. And if it happens that you are an athlete and you need a lot of oxygen for endurance and for muscle function, performance, and if the athlete takes some shots of EPO, they can produce more red blood cells and they can perform better in Olympics or something like that, right? So that is called doping, right? So that's actually illegal for Olympic athletes. And like to circumvent that situation, some athletes decided to store their own blood, and just before competition, they go to their doctor and they get infusion of their own blood like back into them. That's called blood doping, and it's basically not detected. It's not like you're using some drugs; you're just getting your blood back. But it's also dangerous because if you increase your hematocrit count a bit too much, then your blood starts to coagulate on its own, and you can die. So not a good idea. Yeah. Anyway, there's a lot to talk about, the blood. But let's go, back here on how this differentiation is happening. Right. So we know what is a hormone and how it's going around. So now let's see that you have cells that decide to go to the myeloid pathway. Right. So it got a signal from the hormones. So that was the external signal. So now you're going to have internal signals that are going to upregulate transcription factors, right? So two major transcription factors of your myeloid path are these guys over here. So it's PU1 and GATA1. Don't tell me what it stands for. So PU1 and GATA1. The expression of these two factors, from your precursor cell, the expression of these two factors is actually quite low. So this is what you call lineage priming, okay? So at first, the cell is expressing both of them, but then you are going to decide, the cell is going to decide whether it wants to express one over the other. Okay, because depending on whether it's going to express PU1 or whether it's going to be express GATA1, it is going to take further branches out of that path. Okay, so now let's say that you have a signal that will cause, so signal cytokine could be hormone, right, that is going to cause this PU1 transcription factor level to increase. Once these factors start, this PU1 TF start to increase, this guy is going to slow down or is going to stop the expression of the other guy, GATA1. All right. So now this cell got a signal is telling it, okay, increase your concentration of PU1. Once this happens, then automatically GATA1 gets suppressed, and the cell is going to take a path that is going to become, let's say, for example, here, a granulocyte. Okay. So these two transcription factors are actually antagonistic. So if you favor PU1, then you are suppressing GATA1. Now, if instead of favoring PU1, if you start favoring GATA1, that is going to have antagonistic effect on the expression of PU1. So it's like a seesaw here. Okay. If you increase GATA1, then you suppress PU1, and your path is going to go towards red blood cell formation. Okay. So there is a whole check and balance of levels of transcription factors that is going to determine whether the cell is going to go one way or the other way. So here, two master transcription factors of myeloid lineage, right? So you have here your PU1, and PU1 is going to mainly produce your white blood cells, okay? In comparison to your GATA1, that if GATA1 wins, then you're mainly going to produce red blood cells. Okay, so is it going to be one or the other? Okay, is that clear? Yeah. Okay. All right. Now looking here at your lymphoid lineage, all right? So myeloid is that way. Now you're going to go towards lymphoid pathway. You also have hormones that are going to go there and stimulate differentiation, and that stimulation is going to give you other transcription factors, and these two here are the most important transcription factors for myeloid lineage. So you have this guy Icarus, and you have GATA3. Okay, it's not GATA1. Okay, this guy is GATA3. Okay, very, very important for your lymphoid lineage. Right? Lymphoid, what's going to give you? B, T, or natural killer cells. Right? Now, once you start expressing Icarus, okay, on the transcription factor, okay, Icarus is very important for suppressing any idea of becoming a myeloid cell. All right, so once Icarus is there, then this cell is committed to becoming a lymphocyte of some kind. All right. If Icarus is not there, this guy could change his mind and express something else, very something, you know, and become a myeloid. But no, once you express Icarus, this is it. The cell cannot change its mind. It cannot become myeloid. It has to be lymphoid path. Okay. So Icarus is very, very important for myeloid. What are you going to get out of the myeloid? B cells and T cells, natural killer cells. So here I put a cool clinical correlation, okay, that is actually quite detrimental in case you have some problem in the gene that is expressing Icarus. If you have mutations and Icarus is not expressed well, this is one of the cases in which you can develop a type of leukemia, which is white blood cell cancer, okay? So in this case here, what happens is that you get B cells that cannot further develop in the bone marrow. So they get stuck in this primary stage of differentiation, but they cannot really become B cells. Okay, so these B cells get stuck, and because they keep on dividing and keep on getting stuck in this stage, this is how you get the development of that type of cancer, of blood cancer. Okay. All right, so this is situation with Icarus, very, very important. Okay. Now, let's say that you have Icarus and then you start expressing a lot of GATA3 transcription factor. Now, GATA3 is very, very important for development of T cells. Okay, so here, Icarus and GATA3. So you're going to have your B and T cells. With Icarus here, you have a certain differentiation that you go for B cells. But then if you express GATA3, that is really commitment of this development towards the T cell line. Okay? And you do have some other transcription factors that we're going to see when we get to this T cell development. Right? So what you should know here, you should know Icarus commits to lymphoid lineage and represses any thought of becoming myeloid. And then you have Icarus with GATA3. If you have a GATA3 being expressed, you go to T cell. If you only had the Icarus, then you can go to B cells. Right? Cool? No. Okay, so here is a little summary of these important transcription factors. So what we mentioned here, we mentioned GATA1, that is myeloid, important for red blood cells and platelets. You have your PU1, okay, mainly myeloid, this is where I mentioned it, right? PU1 is important also here for B cells, but think PU1 going to myeloid. Then you have here, oh, I didn't really mention this guy here, so forget about that. Forget about this guy here. You have Icarus, right? All lymphoid cells. And you have GATA3 for T cells, right? So 4 important transcription factors here. Okay. All right, so here, big picture, what is in the blood? Right, so you put blood in a test tube, you centrifuge it, right, spin it down, you're going to have separation of blood components. So here is the more dense part of the blood where you get your red blood cells. And if you see the proportion of red blood cell to the entire volume, this is what we call the hematocrit, right? Now, when you look at the liquidy part of the blood, you're going to get the plasma, right? Now, here in the plasma, you do not have cells. And then when you look at this little little differentiation here between your red and your plasma, right? You're going to find a small percentage that you call the buffy coat, and this is where you're going to find all of your white blood cells. Okay? So now let's see what is in the white blood cells, because we're really interested in those, right? So the white blood cells, look at this. You have a lot of white blood cells per milliliter. What is a milliliter? 1,000 of a liter, right? It's a very, very tiny volume. So if you have here a pipette tip, anybody working in the lab and you can use those pipette tips? Yeah. So a milliliter would be like this, just a little tip of volume, right? Very, very tiny. In that 1 milliliter, you can have 5,000 to 10,000 white blood cells present. And this is the normal count. Okay, 5,000 to 10,000 per milliliter, right? So what is the percentage of all different types of white blood cells that you're going to find there, right? So you're going to have neutrophils, lymphocytes. Lymphocytes are your B cells, T cells, and natural killer cells. You're going to have monocytes, you have eosinophils and basophils, and the order of their concentration of the number, it goes in this way. So you're going to have mainly neutrophils. Look at this, the biggest percentage, up to 70%. Then you're going to have your lymphocytes. Then you're going to have these cells that are in very low percentage in comparison to the rest. Okay, so look at these monocytes, like 3%, eosinophils, even less. Basophils, like you can barely even like detect them. It's like very, very low percentage. And how do you memorize the order of percentages in the blood? Do you have mnemonics for that? Yeah. Never let monkeys eat bananas. Right? Everybody got that? All right, so that gives you the more or less the percentage of your white blood cells. Okay. Now look at this. If you have a high count of your white blood cells, so let's say above 10,000, if you have very high count of white blood cells, that means your body is most likely fighting an infection, right? Most likely a bacterial infection. Okay. If you have low count of white blood cells, it could be that your body is fighting some other type of infection, like some type of viral infection, or it could be due to some diseases, including cancers. Okay? So those are important things here. All right. So here, getting into more details on the different types of white blood cells. Right? Looking at the myeloid pathway, you're going to find these cells that if you look under the microscope, you see a lot of dots on the cells. So you call those granules. Right? So some of them can be stained and they look pink. Some other ones, they naturally look bluish, right? But if you see little granules in the light microscope, under the light microscope, you call those cells granulocytes. So here are examples of your granulocytes. All right, so you have the neutrophil, eosinophil, basophil, okay? And you have your mast cell, but then your mast cell is derived from your basophil, I believe. Yeah, mast cells derived from basophils. All right. So how do you remember these guys are granulocytes? Well, there is also mnemonics for that. I'm not sure if it works for you, but it worked for me because if you look at here, B-E-N, right? So that spells Ben, right? Ben happens to be the name of my husband, and he happens to be an accountant. That means he manages the grana. You know what grana is in Portuguese? Money, right? So granulocytes, I mean, I associated Ben with money, so granulocytes is B-E-N, so those are the ones that, the grana with granulocytes. You know what I mean? Well, hopefully it works for you too. Just find somebody who's called Ben and manages money. Now you remember granulocytes. Okay. Actually, his title was like a controller, in these companies, and I'm like, yeah, he's very good at controlling the money, sometimes annoyingly. So, but anyway, a different story, right? So back here to granulocytes. All right. Here's important types, granulocytes. What do you find in granulocyte? First thing, you're going to look at the cells and you're going to say, the nucleus looks weird, very weird. It doesn't look like a little round thing in the center of the cell, right? It looks like this polymorphic, morphic kind of thing. This guy actually looks round. It looks like a sphere, right? But these other guys here, look at this. You actually call them polymorphonuclear cells, okay? Mononuclear. That means it might look like you have 3 nuclei here, but no, it's only one nucleus. It is connected. All these lobes here are connected. This guy here has 2 lobes, right? This one here has a strange looking thing, but it's only one nucleus and it has strange forms, okay? When you look at this cell and it has more than 3 lobes, usually it is a neutrophil, okay? If you look at the cell and it has these 2 lobes, looking like, I don't know, the face of a fly using sunglasses, kind of. This guy is an eosinophil. Here it is. Here it is. It kind of looks like a fly with sunglasses. So let's help it. Okay, here. He's looking at you. He's like, oh, a mouth over there too. All right, so at least this guy has sunglasses. That's your eosinophil. Okay. And look at this basophil. It's kind of strange. You can't really make up what is the shape over there, okay? But once you look at the cells, you should something should come to your mind that they are granulocytes, and then you're going to start associating with the function of these cells. Okay? All right. So what do they do, right? Remember, they have 2 to 5 lobes. That's why I call polymorphonuclear cell. Okay, but remember only one nucleus, right? So here, job of your neutrophil. Remember, your neutrophils are the ones that are most abundant in your blood, right? Up to 70% of your white blood cells. The other name for white blood cells is leukocytes. Okay, so these guys over here, they're patrolling the blood, right? They're walking around in the blood, and they are phagocytic. Okay, so if they find bacteria in the blood, they're going to eat it up and digest it and burst the life out of it. Okay, so these guys are very good at detecting pathogens in the blood. Okay, but they do not have a long life. They have a very short life. They die within 3 days of their production. So your red bone marrow is always producing new neutrophils. Okay, only 1 to 3 days they live in the blood. Okay, now remember, those are the neutrophils are granulocytes, so they have granules that are already packed with enzymes, and they are ready to jump into action. So when they eat something, the enzymes are going to digest something that they ate. Okay, so they have these lytic enzymes, and besides the enzymes, they can also activate their oxidative burst type of function. Okay, in case a pathogen is not killed with the enzymes, which is actually 98% of pathogens that are ingested, then you activate the oxidative burst, and that should kill the pathogen. Okay, so neutrophils are very, very good at that, and they patrol pathogens in the blood. Neutrophils do not go to tissues. They can get out of blood and go to certain areas of inflammation, but their main job is to patrol the blood. Okay. Then you have this other guy over here, these basophils. Very, very low percentage of these cells in the blood, right? So they go around in the blood, they are in the circulation, okay? But look at this, they are not really attacking other cells, all right? They are not very good phagocytic cells, all right? I don't even think they do phagocytosis, right? But what happens here is that they are carrying those granules that are packed with inflammatory stuff, mainly histamine, right? They also carry these other things, they're leukotrienes, okay? So these guys here, they are very important to cause vasodilation. You're going to dilate your blood vessels if those elements are dumped out of the cell, okay? So they're very good vasodilators. Besides that, okay, when the basophil comes out of the circulation and they go into certain tissues, like for example, to your skin, right? Also some other connective tissues. What happens is that they start to differentiate a little bit more. Instead of just being basophils, they transform into mast cells, right? And these mast cells, which is just a derivation of the basophil, they sit in the tissues and they are ready, just waiting for some kind of activation, right? When they get activated, they dump their granules, the contents of their granules out there in the tissue. And remember, a lot of the contents are histamine and leukotriene, okay? So they are going to cause inflammation in the tissue. And you can associate mast cells with allergies, right? When you have allergies, what do you take? What kind of drug? Antihistamine, right? Because you want to block the effect of the histamines. Remember, histamine is also a neurotransmitter, right? Did I mention that in the class before? Yeah. So histamine is a kind of a neurotransmitter. Is it excitatory? Is it a downer or an upper? What kind of neurotransmitter? All right, so let's say you don't know the answer. You have no idea what I'm talking about, right? But let's say most people have allergies. I mean, how many people have allergies? How many of you had to take antihistamine? Yeah. Depending on the antihistamine you took, okay, let's say first generation, the second generation, to me they all were kind of like the same, right? What do you feel after you take antihistamine? Tired, sleepy, right? So histamine is excitatory. If you quiet the histamine in your brain, you get tired, right? So histamine is an excitatory neurotransmitter, right? All right, so mast cells are going to cause inflammation because they dump the histamine in the tissues, right? All right, so that's the job of your mast cell. Then you have these other guys here. These are the interesting fly-looking, fly with sunglasses-looking guys. Okay, the eosinophils, right? What happens with these guys here? They are not very known for phagocytosis, but they can phagocytose stuff. But they are very, very important to attack parasites, meaning things that are bigger than themselves. Okay, so the phagocytotic nature of the cells is, not really that significant because they can't eat something that is bigger than themselves. They can only eat little things that are smaller than themselves, which totally makes sense, right? All right, so these guys here, they actually have very, very short lifespan. They only live for a few hours, okay, like less than a day, at the most half of the day, all right? And they are in a low number in comparison to the other cells in the blood anyway, right? All right, so what happens? Those are the guys that have the granules, and the granules they carry around, those have to be very potent granules stuff, because that's their main defense mechanism against parasites. So they go there, they feel the parasite, they dump the granules, and the granules have toxic stuff that's going to damage the parasite and hopefully kill the parasite. Okay? Unfortunately, that stuff is so toxic that it's going to damage your cells too. Whatever is close to the dumping ground of the eosinophils is going to get some damage. Okay? Hopefully, most of the damage is going to go towards the parasite and the rest of your body is going to survive, but you do have some collateral damage that happens. Okay. All right. So this I took from Lecturio, okay? So give you a little chart of your typical, eosinophil, basophil, mast cell. Here they put the guy with the sunglasses. Now don't get confused. Just go with the other picture. Okay, I mean here the basophil. I like to think of eosinophil as the sunglasses. Right? They fly, they eat the worms, something like that. Okay. All right, so here a bit more on this story. So I do have some, repetition of information, but it's a lot of cells to get used to, right? And they do have a lot of functions. So, the more you get used to them, the better it is for you to really familiarize, right? So that's a bit redundant, right? All right. So the ones that are very, very important for phagocytosis. Here is your neutrophil, right? So neutrophils, they are running around in the blood. So here is your neutrophil, okay? In the blood, you're also going to find your monocytes, right? Now, remember, neutrophils are important to catch a blood-borne pathogen. Now, your monocyte is going to come out of the blood vessel at a site where you have inflammation happening. And it goes to the tissue, okay, it sits in the tissue and becomes a macrophage when it is in the tissue. Okay, so monocytes come out, become macrophages. At least they both are starting with the letter M, right? Macrophage is sitting in the tissue, so the one that is floating in the blood has to be the monocyte, okay? So what happens with the cells? They have receptors on their surface. They are going to interact with sites of the blood vessel that are close to inflammation. So that tells you the blood vessel itself is going to feel when you have some tissue damage or when you have some type of infection going on. And the blood vessel is going to start expressing molecules that are going to interact with those cells in order to call those cells to come out of the blood and go to the site and attack whatever is happening over there. Okay, and yes, we're going to have more of this later. All right. So that was a story with your neutrophil monocyte. So here looking at your basophils, right? Basophils in the blood, mast cells in the tissue, right? These guys, they don't really do the phagocytosis, right? So what these guys do, they have those granules that are ready to be dumped, right? Important granules that they have, those are here, histamine, okay? Histamine, prostaglandins, those two over here are going to cause vasodilation, okay? Histamine is the big one for vasodilation, right? Now, besides that, they also have this other molecule here, this heparin. Heparin is very good to prevent blood clots, so prevent coagulation, okay? And they also have this other effector molecule that is called pyrogen. That molecule acts as like a hormone. It goes in the bloodstream, it travels all the way to your brain, it goes into the hypothalamus in your brain, and it causes fever. Okay, so pyrogen causes fever, right? So besides that, they also have those chemotactic factors that those are going to cause, remember the chemotaxis, they're going to attract other white blood cells to get to the site of inflammation or infection. Okay, so those are your mast cells at work. All right. So here is the picture of your eosinophils. And notice that all these slides I took from these YouTube videos, so I put the source there in case you want to watch the entire video. This guy's pretty good, okay? All right, so these guys, eosinophils, those are the ones that attack the parasites, okay? Those are your little sunglasses. Now here they look like earphones, right? What do you call them, earmuffs or something? All right, but I like to think of them of sunglasses, right? So they do have those granules, they're part of the band, right? They do have those granules. They are going to interact with worms or bigger things than themselves, and they're going to dump those granules to cause damage to those bigger worms. Once the pieces of the worms, they get chopped off, and then if they are smaller than the eosinophil, then this guy is going to be able to phagocytose. Otherwise, it can't eat something bigger than itself. Okay. Important things to know, even at this stage with eosinophils, is that they have receptors on their surface. Those receptors are going to interact with a specific type of antibodies. Okay. The antibodies are produced by B cells, actually by plasma cells, but they're coming from the B cell. So the antibodies are actually part of your adaptive immune system. Right? Remember, the innate and adaptive, they're always talking to each other. They're complementing their functions, right? So here what is showing is that the receptor on your eosinophil surface is able to detect when your antibody has interacted with the parasite, okay? So your eosinophil by itself is not detecting the parasite directly, but here it is interacting with the antibody, and then it is able to dump this stuff at the surface of your worm or parasite. Okay. So now we talk about the agranulocytes. Those are the ones that if you look under the microscope, you're not going to see those granules. Does it mean that it doesn't have granules? No, it means that you're not going to see them. Right? But they also have granules. You just can't see them in the light microscope. All right? So agranulocytes, they're everything else but the band. All right? So you have your monocytes. Remember, monocytes become the macrophages. Right? You have macrophages. You're going to have these guys, dendritic cells. Very, very important guys. Right? And then you have these other precursors here, that those are called megakaryocyte, erythroid mast cell progenitor. Okay, there is a long thing for those progenitors. All right, so the main progenitors, they are going to become different cell types. So you're going to have here this type of megakaryocytes that make platelets right here. Then you have precursor for your red blood cell, and then you have precursor here for the mast cell. Okay, so they're coming from the same type of MEMP progenitor. Okay, so agranulocytes, those here and those over there. The mast cell is coming from the same progenitor that is going to become either megakaryocyte or red blood cell. This diagram is not very clear, so let's put a progenitor right here. Oh, I know what is missing. Wait. This guy here, myeloid progenitor. Okay, let's take a little detour right here. This myeloid progenitor becomes this MEMP, okay, this cell here. Now this cell here can go into this way, that way, or this way, okay? This diagram was not very accurate, okay? So mainly these other types of your myeloid pathway that are going to be your agranulocytes. Let's look more into them here. All right, so your agranulocyte. Here you have your monocyte. Okay, the shape of your nucleus here is like a kidney bean, usually, right? So you have your monocyte. When it goes into the tissue, it starts to have those pseudopods kind of projecting out of the cell. So those are your macrophages, right? Compare here to the star-looking cell that is your dendritic cell, right? So dendritic cells are the very, very important cells. They're in between the two immune systems, right? And also with the agranulocytes, you have those megakaryocytes or the MEMP precursors. Okay? All right, so here, functions of these guys. Just focusing here on your white blood cells or your leukocytes. Right? So first thing you have here is monocyte. They go to tissue mature, they become macrophages, and they're very, very important eaters. Those are your very important phagocytic cells. Right? When they go into the tissues, they sit there for a very, very long time. So they have a long life. Okay. So those are your monocytes. Then here you get your dendritic cells, right? Those are everywhere in all the tissues. You're going to find them especially in your skin, like in live portion, right, deep skin, and you're going to find them a lot in the mucus. So every organ that is in contact with the outside environment, like your lungs and your GI tract and your genitourinary tract, those are going to have dendritic cells in the mucus layers, okay? Those are the ones that are very, very important interactions between adaptive and innate immune system, because those are your professional antigen presenting cells, right? You have other cells that are antigen presenting, like for example, B cells, okay? But dendritic cells, they are much better. They are professional. They went to school only to become presenting cells, right? So dendritic cells are really the important one, okay? What do? They are going to get antigen, they're going to get a pathogen, they are going to travel from the tissue all the way to a meeting place that is a secondary lymphoid tissue, and they're going to present a piece of that pathogen. Sometimes they present the whole pathogen, but let's say a piece of that pathogen, they're going to present it to a naive T cell. They're going to make the naive T cell become not so naive, okay, become mature, okay, start to mature. They are going to present to the T cells, and they are also going to present pathogens to the B cells, okay, but then it's a different type of interaction. To the B cells, actually, the dendritic cells bring the entire pathogen. They do not chop up the pathogen. They just bring the entire pathogen and they serve it to the B cells. Okay, this is also a function of the dendritic cells, okay? Which is different than these other cells here. They're called follicular dendritic cells, right? Those cells, they are not in the rest of the body. They are only found in in secondary lymphoid tissues, meaning lymph node or spleen, okay? Follicular dendritic cells, they only have one job. They catch pathogens that get to those tissues, and then they present the pathogens to the B cells, and they make the B cells to work better, okay? So those are all parts of your agranulocytes, all right? Besides those cells here of your innate immune system, so these are all innate, right? Then you have your lymphocytes. That is your adaptive right here. Right? Lymphocytes. No, I almost made a mistake here. Okay, let me delete this. Okay, here we go. Your adaptive are your B cells and your T cells, right? So lymphocytes. Natural killer cells are also lymphocytes, but that is innate immune system. Okay. All right. So here is exercise for you to do it on your own, all right, to match the function of the cells to the name of the cells. And finally, you get at least a couple of points or 3 points out of the class. So we're going to say here, get your points, get your eye clickers. And the question is, what is the job of your eosinophils? In your votes. And that's the part when you can cheat, you know, talk to your neighbors. Go back on your notes and see what those guys are doing. It's not working. Okay, that's bad news. Let's try it again. I'll leave it up some more. For which one? For which cell? Follicular dendritic cells are only found in the lymph nodes and also in the spleen. Yeah. Okay, you have some people that are not giving me the right answer here. Okay, I'm going to stop it. Does anybody need more time? Yeah, okay.
Just very slow.
Okay, well, hopefully it will go in. I'm going to stop here, and if you did not get the credits, you'll see me after the class. What is your answer?
C.
Yay! C. Okay. So some people answer A, and I see a bit of confusion here because I mentioned that they're not really good at phagocytosis because they cannot eat something that is bigger than themselves. But if they chop the thing down to smaller pieces, then they can do the phagocytosis, right? So it's not that they are totally non-phagocytic, just that they can't eat something bigger, but if they have small pieces, then they can eat the small pieces. This is why A is not correct. All right, and here it is, guys, the dendritic cells, right? Very, very important cells. You have a precursor dendritic cell, okay? Dendritic cell, you mainly have two important types here of the dendritic cell, right? They're coming from the same precursor, and then you have a third one that that is important for information. So the first one, okay? First thing to know about the dendritic cells, you know they're coming from the myeloid pathway, right? Because it's not B or T cell or natural killer, so it has to be from myeloid pathway. So the first one you see here, your classical dendritic cell, okay? These guys here, they like to hang around the skin, okay? So connective tissue, they hang around the skin, and they like to catch pathogens that have crossed the skin barrier, right? And if they catch it, they will eat it up, and they will travel through the lymph, through the lymphatics, and they're going to go to a meeting place that is a lymph node, and they're going to present a piece of that pathogen to the T cells. So this is the job of your classical dendritic cell, right? From the skin, from the tissues, and then catch it, and then go to the lymph node and present to tissue cell. Then you have this other type here, dendritic cell, the plasmacytoid dendritic cell. Okay, those guys, they really go around in the blood. Okay, they have one important job, that is the production of interferon, and interferon is the first line of defense against viruses. Okay, so these plasmacytoid cells, dendritic cells, they are important to fight viral infections or viruses that they're running around in the blood, or blood-borne pathogens. Okay, so they're running around in the blood, they catch a pathogen, and they do the whole thing to go to the lymph and present to the T cell. Okay, and then you have this other guy here, those other guys that are inflammatory dendritic cells. Okay, so those guys here are very, very important to work in inflamed tissues. Okay, these guys here, they might derive from that monocyte. Remember, monocyte goes to tissue, become macrophages. Now they could also become inflammatory dendritic cells, but I like for you to think of monocytes really becoming macrophages. You go to the tissue, they become macrophage. That's a symbol for the macrophage. Okay, becoming inflammatory dendritic cell is not the main job of monocytes. All right, so now talking just a little bit about the guys that are going to become your lymphoid cells. All right, so you have here your common lymphoid progenitor cells, and they are going to become either B or T or natural killer. All right, so here they are, B and T cells or natural killer cells. Okay, now you do have a certain variation of a T cell that is called innate lymphoid cell. Okay, so when you get to differentiation of those natural killers or innate lymphoid cells, then we're going to mention more about that. But mainly BT, natural killer, that's pretty much it. Now, how are you going to differentiate whether these lymphoid cells, whether they're going to become B or T cells? It all has to do with the receptors that they have, with the markers that they have on their surface. Okay, so here are your lymphocytes, right? You have a progenitor that can become one of these three. Now here we go. When you have a progenitor that is going to become a T cell, then they're going to have surface markers that are going to be the CD molecules, and they're going to have either CD4 or they're going to have CD8 on their surface. Besides that, they're also going to have a T cell receptor, right? So T cell, they're going to be either CD4 or CD8. If your cell is going to become a B cell, now it's going to have a B cell receptor that is also known as an antibody, but it's also going to have some important surface marker that is not here. So let's put a surface marker here, and this guy here is called CD19. Okay, CD19. That's important B cell surface marker. Okay. All right. Now, when you look at your natural killer cell, it doesn't have those CD markers. Okay, it has something else. But your natural killer cells, they will have, I think, a CD. I forgot the number of that CD. But it does not have B cell receptor. It doesn't have B cell. It doesn't have T cell receptor. Okay, it's just an odd-looking type of lymphocyte. to see how they work too. All right. So here, yes, I know it's a lot of information in this slide, but keep in mind that your T cells, they are going to have important markers. They're either going to be CD4 or they're going to be CD8. If the marker is a CD4, then they can become all different kinds of T cells. They're going to do all different types of jobs. Okay? So when the T cell has a CD4, they are going to interact with antigen that is presented by a professional antigen presenting cell. What comes to your mind when I say professional antigen presenting cell? Dendritic cell. So the dendritic cell is going to have on its surface this presenting thing that is called MHC2. Okay? CD4 interacts with MHC2. After it interacts with this MHC2, then the T cell can become some kind of helper cell, or it could even become a regulatory cell. Okay? On the other hand, if your T cell has a marker that is a CD8, then this marker, this type of receptor, is going to interact with another presenting molecule that is called MHC1. Okay? Dendritic cells also have MHC1. Okay? So MHC1 interacts with CD8, and after this interaction, you're going to activate your CD8, and they're going to become other subtypes of CD8s. CD8 T cells, those are called killer cells too, but they're killer cells of your adaptive immune system. Okay? So you call them cytotoxic T cells. They kill. Okay? They're not like the helper T cells. Helper T cells don't really kill much. They help other cells to do the killing, right? But cytotoxic, they do the killing. Okay? All right, so just to summarize here on your I'm running out of time. Okay, so I'm going to catch up from this slide on Thursday.